Hello and welcome back to the biological molecules chapter. This is video 5 where today we're going to be discussing proteins. Firstly, let's talk about the general structure of proteins, focusing on amino acids and how they link together. First, amino acids are the building blocks of proteins. Each amino acid has a central carbon atom, known as the alpha carbon. This central carbon is bonded to three groups, an amine group, a carboxyl group, and a variable R group. There are over 20 different amino acids, each with unique R groups, which give amino acids distinctive properties. Now let's talk about how these amino acids link together to form proteins. During protein synthesis, amino acids are joined through a process called condensation. In this reaction, the hydroxyl group, the OH, from the carboxyl group of one amino acid, and a hydrogen atom, H, from the amine group of another amino acid combine to form water, or H2O. This release of water is crucial for linking amino acids. The actual bond formed between these amino acids is known as a peptide bond. It connects the carbon of the carboxyl group of one amino acid to the nitrogen of the amine group of the next amino acid. This linkage creates a chain of amino acids, which is called a polypeptide. As more amino acids join together through peptide bonds, they form longer chains, eventually folding into complex structures. These structures are essential for the diverse function of proteins in living organisms. So just to summarize this slide, proteins are made up of amino acids, which are connected by peptide bonds through condensation reactions. The unique properties of proteins come from the sequence and combination of the 20 different amino acids. So now let's discuss the primary and secondary structures of proteins. The primary structure is all about the sequence of amino acids. Imagine a long chain made up of different beads. Each bead is an amino acid. The order in which these beads or amino acids are arranged is crucial because it determines how the protein will fold and function. Following that, we move on to the secondary structure of proteins. This is where things get a bit more complex. In this level, specific interactions between atoms come into play. Each amino acid has a backbone with groups called NH and CO. The NH group has a slightly positive charge, while the CO group has a slightly negative charge. Because of these charges, the NH group from one amino acid can form a hydrogen bond with the CO group of another amino acid. These bonds create a twist or a fold in the protein chain. The main two shapes that this can lead to is the alpha helix, which is a sort of spiral staircase formation or shape which the protein takes when it twists into a helical form, or the beta pleated sheet. This is where the protein chain folds back and forth on itself to create the sheet structure. These secondary structures help the protein to achieve its final functional form, which is essential for its role in the cell. So now we will move on to discuss tertiary and quaternary structures of proteins. Following on from the secondary structure, the tertiary structure further folds into a more intricate 3D shape. This final shape is maintained by several types of bonds, the first being disulfide bridges. These are strong covalent bonds formed between sulfur atoms in the R groups of certain amino acids, providing stability to the protein structure. And secondly, ionic bonds. These are weaker bonds that occur between positively and negatively charged groups on the amino acids that are not involved in the peptide bonds. The final form that a protein can take is the quaternary structure, where in some proteins multiple tertiary structures come together to form a functional protein. These polypeptide chains interact and are held together by various bonds, including disulfide bridges, ionic bonds, and hydrogen bonds. The final section of this video will be the test for proteins, or the Biuret test. There are 4 steps to this test, and it allows us to test for the presence of protein within food samples. So the first step is to put the food sample into the test tube. We will then add in sodium hydroxide, NaOH, and mix thoroughly. Following that, we will then add copper 2 sulfate, a few drops of this, and mix again. The resulting mixture in the test tube, if a protein is present, will turn purple. Remember, P for purple, P for protein. If no protein is present, the mixture will remain blue. Thank you very much for watching this video. Hopefully you found it informative and useful for your A-level revision. If you enjoyed it, then make sure to like, comment and subscribe, as it means a lot to the channel and helps us grow. Also, share this video with your friends and feel free to check out any other videos on this channel as well.